Gene is a piece of the DNA we call blueprint for life. DNA or gene gives instruction that goes to the mRNA, then mRNA translates to the protein.
The protein pretty much responsible for all our biological functions. That's how we work because of our protein. Our gene determines which protein to be expressed.
Every cell you have to have a sort of DNA to make the cell function.
But cells in different organs, different tissues, they express certain subset of genes.
The genetic variant can cause called a personal trait, which is neutral or benign.
But some of them are crucial for our health.
If that particular variant end up in a very critical part of a gene,
we may produce a faulty protein, or we cannot produce a specific protein at all.
that became disease-causing. Oftentimes, we call mutation.
For some genetic disease, the cause could be very complicated. Without understanding the mechanism, it's certainly very difficult to design any treatment.
Gene editing can modulate or correct a gene which you believe in causing disease.
It's a very powerful tool which could be used to produce the right form of protein, which responds for most biological functions.
Before, there is no tool can do this job very, very efficiently.
Even somebody carries the genetic variants, this genetic variants may not affect all cells or tissues.
When CRISPR coming, so this became a reality. So you can editing or correct the gene very efficient way.
Basically, you can target and edit specific cells. So there's a lot of advantage makes CRISPR so revolutionary. And then now a lot of people, including us, try to use this technology to treat genetic disease. Pretty much the biological structure of the brain makes drug delivery to the brain. In this case, deliver our genome editing to the brain. Very, very challenging.
This very innovative technology, in the short we call STAP, allow us to deliver the CRISPR editing more efficient to the brain.
Basically, it's a technology based on the use of small molecules to deliver protein-based genome editing machinery. We have developed thousands of STAP molecules already. So we screen those STAP molecules. We found certain STAP molecules like a specific cell type. They are enable deliver the CRISPR machinery into the brain and disperse them throughout the brain. So that's very unique.
We have 5,000 diseases, we know the mechanism, but each one may have a different mechanism. So therefore, individual disease, you may have develop individual sort of for the tool, even under the umbrella for the CRISPR editing. So far, I think we use the Cas9 for the disease we're working on right now is Angelman syndrome that potentially can apply to a few hundred other neurogenetic diseases.
Our hope is that in the next few years, with the support from the NIH, we can move this STAP-based therapy into clinic. But the STAP technology is a platform technology, which means that we can potentially adapt this technology for deliver our other payloads to other cells and tissues. That means in long term, we hope this technology can be repurposed for treatment of many other rare diseases.